Hi, I'm Jay Phelan. Let's talk about biology. I'm going to talk today about inheritance of traits that are influenced by 2 or even more genes. And we refer to this whole area as extensions to Mendel or modified Mendelian ratios. And there are several different permutations of this. This will be the first of a few of those, and it's on a topic that we call complementation. Now, recall that when Mendel was first exploring inheritance, why does its offspring resemble their parents more than some other individual in a population? He looked at a lot of different traits, 7 different traits, but he was always looking at physical characteristics that were coded for by a single gene that had 2 alleles, one of which was completely dominant over the other. There were no environmental effects, and so on. This was great for deducing how the laws of heredity worked, but there are a lot of traits that don't satisfy those conditions. So we have to ask the question, is every trait influenced by just one gene? No, of course not. Many are, whether we're looking at small organisms or humans. But it's clear that if we want to get a rich, full understanding of inheritance, we need to be able to expand our Mendelian genetics model for thinking about it into something that will accommodate a trait that is influenced by more than one gene. So suppose that you are working on an organism, maybe mice, yellow-necked mice here, or maybe you're working on cats. We've got a cat here. You see the funky bent ears. We're going to talk about that. Or maybe you're looking at dogs, or maybe you're looking at fruit flies. Some model organism that you're looking at, and suppose that every now and then you see animals that have an unusual mutation. That is, they show bent ears. And it could be anything, but we're going to illustrate it with this example. So they're born, and unlike all the other animals that have what we would call the wild type, the more typical one, the common one that we generally see, we see this other trait. So you get these unusual individuals, and then through standard crosses, you figure out that this condition is inherited as a single gene autosomal recessive trait. Now, don't just take my word for that. Imagine, how would you do that? When I say standard crosses, what do I mean? What would you do? How would you conclude that? If it's autosomal recessive, think about it, right? They'd have to have 2 copies of the recessive. So what would happen if you mated together 2 of the individuals that had the bent ears? Think about what the offspring would have to look like. Anyway, so you should be able to figure out that, but we'll go from there, that it's inherited as an autosomal recessive trait. Now let's say you've got a friend, a biologist on the other side of the country, and that person is working on the same model organism, let's say these mice, and they also notice every now and then, we get the unusual mutation with the bent ears. And they do the same crosses that you've done to ascertain that it's inherited as an autosomal recessive condition. We call this process where you identify different phenotypic expressions of mutations a mutant screen. And all model organisms will have a whole library of different things we know about them based on mutant screens. So you've got your bent-eared mice. Your friend on the other side of the country has their bent-eared mice. You both know that they're autosomal recessive. What's your first assumption going to be about the genetics of this trait? Well, we like to use what we call parsimony. Just the simplest explanation is typically the one we're going to assume is correct until we have reason to believe otherwise. So you would just say, all right, it's probably the same mutation, probably the same thing in both populations that is causing it. There is one gene, and if you have an autosomal recessive, you're going to have the bent ears. Could you test that hypothesis? Sure. What are you going to do? We've got bent-eared mice from your lab, bent-eared mice from your friend's lab. Do what any good geneticist would do. You're going to cross them. Now, think about it. We said it was autosomal recessive, so we should know the genotype of both of those. So you cross them. What are you expecting? You're expecting that they're both, little a, little a, little a, little a. So for that trait, that's going to be all that's possible. And yet, when you do this cross, every offspring has normal ears. What's going on? That's odd. That's unexpected. So now, that assumption we just made, all right, same mutation, and all the animals with bent ears have autosomal recessive genotype for that trait. That doesn't work here. This observation tells us we cannot use that as our hypothesis for what's going on. So what would you do now? Like any good geneticist, you'd say, well, let's cross those. All these normal-eared mice that came from the cross of bent-eared from two separate populations. So you cross the F1 mice, and I've illustrated here the crosses we had. So the initial P generation for parents, bent ears, you should be able to write down what the genotypes for those, what we thought they were. But then we got all normal ears. Now that tells you whatever you wrote up there, probably not correct. When you cross all of the normal-eared ones, the F1s, now you get the F2 generation, you get about half and half, actually slightly more of the normal ear phenotype, but you still get a lot of the bent ear, 9-7 if you collect a large enough sample size to calculate a ratio. You say, all right, 9-7. What does that tell us about the inheritance pattern of this bent ear trait in these mice? Well, you look at 9-7, obviously it's a modification of 9-3 to 3-1. I'm kidding, it's not obvious. But I'm telling you that it's obvious. And do you remember where 9-3 to 3-1 comes from? If you did a Punnett square, where you looked at two traits simultaneously. When we looked at Mendel looking at independent assortment. So you look at two separate genes at the same time, and you put 4 different gametes on one side that the father has, 4 different gametes with respect to these two traits on the other, and you have all the different possibilities. 16 squares of genotypes that can come together, and they end up being a ratio 9, where you show at least one of the dominant dominant traits, and so on. I'm going to go through this in detail. So here's the cross: big A, big B, little b, big A, big B, little b. So we call it a double heterozygote for those two traits. When you do that, because that first individual on the left side there, big A, big B, little b, think of the gametes that it can produce. Mendel's law of segregation says it's only going to get one copy of each gene. So it's going to get maybe the big A and then one of the B, maybe a big B. So big A, big B. Or big A, maybe little b. There's another one. Or the little a and the big B. Or the little a and the little b. So there are 4 different options of the gametes. Same thing for the other one. So you do the Punnett square, and you'll find out that 9 out of 16 have a big A and something else. I put the line there as a placeholder. It doesn't matter if it's big A or little a, because you have the dominant allele. You're going to show the phenotype for that dominant trait for A. Same thing for B. You have at least one big B. The other one might be little b, might be big B. Doesn't matter. You're going to show phenotypically A and B. In 3 out of the 16 times, you're going to inherit at least one big A. The other one you don't know, could be either, but it's going to show the big A trait. And then for B, you're going to inherit 2 little b's. You've got the recessive condition for that one. You're also going to have the reverse of this. In 3 out of 16 cases, you're going to inherit 2 of the little a's, one from the father, one from the mother. And then for the B, you're going to get at least one big B. Maybe it's big B, maybe it's big B, little b. And then finally, 1 out of 16 times, you're going to get 2 recessive alleles for A, 2 recessive alleles for B. So there's our 9-3 to 3-1 ratio. This happens anytime you cross double heterozygotes for 2 genes. Now, at least once in your life, I know you don't want to do this, at least once in your life, go through the tedium of the 4x4 Punnett square, where and it's not 4 genes, it's 2 genes by 2 genes, but you look at 4 gametes, and you write out what are the genotypes that would result, because each one of those is equally likely, and then figure out from the 16 combinations you have there of genotypes, what phenotypes would they be, given that you have complete dominance. That's where you find out, oh, in 9 of the cases, they show the dominant for both. In 3 of them, they show it for A but not B. In 3 of them, they show it for B but not A. In 1, they show it for neither of them. So just do it once. Right now. No, later. So in our cross, we had double heterozygotes. We're just throwing this out there as a hypothesis. We got 9 normal, 7 bent. So we look at our double heterozygote cross here, and we think, all right, I see the 9 on the left side there, and then the 3 and 3 and 1. So I think that those phenotypes, the 9, those are going to be the 9 normal, meaning that as long as you have a dominant allele, at least one dominant allele for each of the 2 genes, then you're able to develop the normal ear phenotype. On the other hand, in the 3, 3, and 1, in each of those, the common thing is for at least one of the genes, they're homozygous recessive. And when that's the case, you end up with bent ear phenotype. So that's how we see it. We can propose a biochemical hypothesis for this. We could ask the question, well, why did we get these normal mice from the cross of the mutants? Why did the cross of the F1s give us some of each? So our hypothesis here is that there's two genes influencing this trait, and in your lab and in your friend's lab, it was a mutation in the different gene that was responsible for the bent ear. So in yours, it was the first one. In the others, it was the other one. So they had the same phenotype, but it was caused for different reasons. Here's how it could play out biochemically. And this is true for organisms building up their bodies for almost any trait you look at. You can look at eye color in fruit flies, or you could look at coat color in guinea pigs, or whatever. That you start with something. Maybe this is something you eat in your diet, but it gets modified into something else, compound B. And it might get modified then into something else, into something else, into something else. To make eye color in fruit flies, it's about 20 separate steps. But here, to make the normal ear shape, could just be two. Compound A gets modified to compound B gets modified into the normal ear shape. When we look at biochemical pathways, every step will always be catalyzed by an enzyme. So you need enzyme 1 to go from compound A to compound B. You need enzyme 2 to go from compound B to the normal ear shape. And every enzyme is going to be coded for by a gene. That's usually what they do. So now we've got gene 1, gene 2. This helps us then to understand how we might get this pattern that we saw. If gene 1 or gene 2 were mutated, what happens? So look, let's say that gene 1 was mutated, meaning that the individual inherited homozygous recessive condition. So neither copy of the gene produces enzyme 1 in a way that it works. It can't catalyze the conversion of compound A to compound B. So what happens? Well, we're not converting any of it, and so compound A just builds up. There is no compound B, can't get turned into normal ear shape. We never get there. So you go, bam, gene X can't make enzyme 1, bam, enzyme 1 doesn't exist, bam, compound A can't get converted into compound B, and if you don't have that, you don't have normal ear shape. That's if gene 1 is messed up. On the other hand, gene 2 might be messed up and gene 1 is fine. If that's the case, it doesn't carry the proper instructions to make enzyme 2. So your body doesn't have enzyme 2. So you get compound A somehow, maybe you eat it. You have gene 1 producing enzyme 1, and it catalyzes compound A getting turned into compound B, but now we're stuck. We can't modify that into the normal ear shape. However, we do that, so gene 2 doesn't make enzyme 2. Enzyme 2 then isn't going to exist. We can't catalyze that reaction, and we don't get normal ear shape. In both cases, we end up with the same outcome, phenotypically bent ears, but we do it in different ways. So we call something a complementation test when we do what we did here. We take your mutant individual from your lab, your friend's mutant individuals from their lab, and your first assumption is one trait, two alleles, maybe it's homozygous recessive, but that they're the same. But you cross them, and if when you cross them, you end up with the normal phenotype, wild type phenotype again, then what you have to conclude is, all right, it looks the same, but it's being caused by a breakdown at a different point in the biochemical pathway. So this complementation test is going to tell us that. In our cross here, you could think of, we called them A and B before, but now let's say that there's one gene and it's B for bent, and there's another one for F and for folded. So if you're homozygous recessive for the B gene, then you're going to have bent ears. If you're homozygous recessive for the F, you're going to have folded ears. Same thing, it's going to look like mutant. So our initial cross now was this. You assume that mutations are rare, so that for the gene that we're not looking at, this mutation, the other little b, we assume that for the other one, big F, that they're probably almost everyone in the population is homozygous dominant. But that individual would have bent ears. Big B, little f would also have the bent ear. So we crossed those, and when we did that cross, think about the gametes that they have. The one on the left has a little b in every gamete and a big F in every gamete. That's all it has. The other individual has a big B in every gamete and a little f. So when those come together, you're always going to get the same thing. You're always going to get big B, big F, little f. So all of the F1s are doubly heterozygous. But that's what we did then. We did the double heterozygous heterozygous cross. That's the 4x4 Punnett square that I said you have to do at some point. And we look at it. So I've just replaced it here with B's and F's so you can see it. But 9 out of 16 are going to have at least one dominant. 3 out of 16 are going to have one dominant for B, not the other. 3 are going to have one dominant for F, and so on. So we look at these, and anything that has little f or little b is going to be blocked. That biochemical pathway can't make it all the way down from the beginning to normal ear phenotype. Therefore, anytime you have two recessive alleles, then we're going to end up with a bent ear phenotype. In this case, that would be the 3, and the 1. So 7 out of 16 are going to look like that. 9 normal to 7 bent, and now we at least can understand that that ratio is a red flag. It says, it's sort of like, it adds up to 16. We can see 9 to 3 to 3 to 1 there, probably 2 different genes. And then you can go down the pathway to figuring out what the inheritance pattern is. So we suspect complementation anytime you cross individuals that both exhibit a mutant recessive phenotype, and yet somehow in the F1 generation, we see all wild type phenotypes. That's it. Here's a field of flowers for no reason.